Well, good afternoon, everyone. Welcome to the Soil Health Nexus Cafe series. We're pretty excited today to have Hava Blair with us, graduate student from the University of Minnesota, Department of Soil and Water and Climate. She's going to discuss with us today sharing soil health data with reproducible reports. I'm Paul Gross. I'm an extension educator in Central Michigan, part of the Nexus, along with Christina Carell and Leslie Johnson, who are kind of your co-hosts today. So welcome, Hava, and please start when you have the opportunity. I'll quit sharing my screen and you can share yours.
Okay, well, good afternoon, everyone. Thanks, Paul, for the introduction. Like you said, my name is Hava Blair. I'm a graduate student at the University of Minnesota, advised by Dr. Anna Cates. And today I'm here to share a little bit with you about some soil health research that we've been doing at the University of Minnesota, and specifically focus on some decisions that we've made and a framework that we're using to share some preliminary soil health data with our cooperating farmers using a tool, a software programming language called R, and the R Markdown format. So I just want to do a little housekeeping as we get started. I do have the slides that I'm using today available for download and some of the code that I'll be showing examples of. So if you'd like to follow along or you'd like to see a PDF copy of the reports as I'm going through them today, just head to this link and I'll take you through quickly how you get there. It's following this link and you'll click on the blue button. And then you'll see my slides at the bottom as a PDF, but there's also other documents here that you're free to download and use. And I just want to show you what that looks like in case you haven't seen GitHub before. So you'll click on what you want to download, and then there's a download button there once you're in the file that you'd like to have. So feel free to go there and grab the slides now or later. That website will stay up. But with that little bit done, I think the big goal for today is just to share our strategy and the tools that we use for communicating data about soil health, specifically to our farmer cooperators. So the presentation will take place in 4 main parts. First, I'll just give you a broad overview of our research project, what we're up to, and then I'll talk about what really motivated us to use this strategy of trying to create reproducible and automated reports and how we did that. And I'll focus on the technology that we used and try to convince you that this might be something that you should try in your role as an educator or a student or really anybody that is handling data, doing data analysis, and doing some level of reporting. So I'll go through some details about how to use R and RStudio on a high level. And then finally just end with some questions and some of the things that we've been thinking about on our team about what is the value and the best way for us to be communicating this data and sharing it out as with people in a valuable way. So here in Minnesota, our research is focused on getting a better understanding of soil health management systems on farms here with a focus on cool climates and heavy soils, because those are both really relevant to the farmers in our region and some of the challenges that they experience with adopting things like cover crops and soil health management. And our broad project has 4 big objectives. The first objective is a big data collection effort. So we're collecting soil health data statewide using standardized methods across a whole swath of the state, and I'll show a map in a minute. And then once we've collected that data, the other big thing that we're working on is harmonizing it with other regional data sets from previous studies that have been conducted here in Minnesota. So we're making this effort to collect data, but also aggregate it and harmonize it so we can really be a regional soil health data hub. And in doing that, one key component is developing an actual database. So getting the infrastructure in place and getting everything prepared so that we can have this soil health data accessible and with a public interface that will allow people to explore and sort of see some of the patterns that we're observing as we get this soil health together, this data together, and analyze it. And the final goal, but probably the most important, is that all this work is really pointed at increasing capacity for knowledge sharing amongst farmers that already are operating with soil health management systems. They have the expertise, and getting them connected to other folks that are interested in adopting those systems and working out how they can share and create a social network around adopting soil health management. We are really lucky in this project to work with a number of different partners and cooperators. So we have 27 different farmer cooperators in 6 different counties all across the state, as you can see on the map here. And then we also have some really great connections with our local government, especially the soil and water conservation districts, which are a county level conservation organization here in Minnesota, and then some state government and nonprofit partners too. So we've been able to utilize a lot of local expertise in developing this project. And I just want to talk a little bit about our research design so that as I go through some of the data today, you have a picture in your mind of what we're working with here. So broadly, we're aiming to capture changes in soil health in this data and the variability on a couple different levels. And the first level is really this management level of how can we use these soil health indicators to understand differences due to management when people are using soil health management or conventional management. And then we also want to get an understanding within a field and within sort of a understanding different soil types, what are the differences that we see in these indicators based on hill slope position? So more of a understanding that topography and how that impacts them. And that's closely tied with this concept of a soil map unit that many of you are probably familiar with. We also want to try to get an understanding of more of a baseline or what these soil health indicators would look like in natural areas that are undisturbed. And for us, we're really defining natural area as a fence row or as a tree line, just something to give a contrast to the cultivated land that we're sampling as part of our efforts. And then finally, we are taking advantage of the fact that Minnesota and most states have a variety of different soil types and regional differences in soil. And so we have 4 main regions in our study, a northwest, a central, a southeast, and a southwest. And those can be grouped pretty clearly into distinct broad patterns of soil type and texture and climate. So we're also interested how big those regional differences might be as we're undertaking a sampling effort to measure all the same indicators on all these farms across the different regions. So this is a bird's eye view to give you an idea of what sample collection looks like for us. So you're going to look at 2 fields here, split by a tree line that's sort of a third of the way down there. We have a field that is conventionally managed at the top, and then a field that is managed with a soil health management broadly at the bottom. And for our study, because we're working on working farms, it's not a tightly controlled, plot study. We really have pretty broad definitions of what we mean by soil health management and conventional. By conventional, we are defining it as annual tillage and then shorter, less diverse rotations, usually a corn and soy or corn on corn. And then for soil health management, we're working with people that have been no-tilling for 2 decades, no-tilling for 5 years. There's sort of a range, but it all includes things like no-till or reduced tillage, incorporation of cover crops into the rotation, and diversified rotations. So that's how we're really grouping together the soil health management. So you see I have different colored squares here that represent the 2 different map units, the upper map unit and the lower map unit, so getting at some of that in-field variability. And then in the middle, we would take samples along the tree line there for our natural area samples. And that's just a picture of what it looks like for our sampling effort in each pair of fields. And overall, throughout the project, just to give you a sense of where we are, we're about halfway through. We started in late 2018, early 2019, identifying our cooperators and getting all of these sites organized. We've completed 1 year of sampling in the fall and gone through lab analysis in the winter and still ongoing. And just this spring, within the next couple weeks, we are starting to take infiltration measurements on all these farms. So that's really the first half of our project, and then we have another year's worth of soil collection and infiltration measurements before we'll have a full data set and finish off with some field days to share what we've learned. To give you an idea of the types of indicators that we're measuring, today I'm focusing on sharing a little bit more about the ones on the left under done. So we did soil organic matter, potentially mineralizable nitrogen, 24-hour mineralizable carbon, which some people call soil respiration or burst test, and then a standard nutrient workup of pH, phosphorus, and potassium. You can also see here that we have a number of other indicators that we will be measuring, and they're in progress in the lab or coming soon, kind of on deck. So I won't read all of those, but they serve as a reference to give you a picture of the idea that we're really trying to measure many different soil properties and get an understanding of the relationship between some of these indicators and the management and soil conditions that we're seeing across our sites. And all of this is really to set us up for the big question of the day, which is, for our team, what's the most effective way that we can share data back with the farmers that are giving us access to their land and really making this research possible? And what's the most valuable way to do that? And I think a question that we ran into that really motivated the idea of sending out preliminary reports to farmers was, we weren't totally sure what was most valuable. Is it having a complete data set, which we know is going to take us 2.5 to 3 years? Or are people going to appreciate just seeing a glimpse of things as a way to stay in touch with us and really know what our team is working on? So we decided the latter was valuable, and we wanted to use these reports as a way to share our data, but also just to stay in touch and keep that engagement with the people that we're working with. And as the grad student charged with making these reports, one of my first things that I had to think about was, okay, we have all this data, we have all these cooperators, this is all good, we've got a nice data set. But if you start thinking about a lot of the number crunching involved with creating individualized reports for 27 different cooperators, you quickly come to the conclusion that that's a lot of graphs, right? So we have 27 different farms, 12 different indicators, 2 different map units within each field that we're measuring. So even if you're perfect and you make every graph perfect the first time, that's still more than 600 graphs that you're working through and arranging. And if you're like most people who started working in Microsoft Excel as you did data analysis, you're probably in a workflow where you'll make a graph, do some data analysis, kind of tweak it, get it right, and then copy paste into whatever your final document is. But in our case, that was simply not an option with the number of graphs that we knew we had to make. So we knew we had to come up with a different solution that did not utilize quite so much copy and pasting and allowed us to really iterate on these reports, come up with a template that was going to work for us, and then use that to crunch all this data for our different farmers. So that's what I'm going to talk about next, is how do we, on the technical side, how do we actually do this, and how can we create a template that'll help us with the data analysis and with the presentation all in one package. And just to reiterate, we had these goals for our reports. We want to share the data, we want to keep in touch with farmers, and we also want to show them, what are some sort of interesting patterns that we're seeing just regionally and on their farms, even if we don't yet have a totally complete data set or analysis. We wanted to share something, and we needed to find a way to do that efficiently from just a standpoint of labor and getting the job done. And now that I've been talking about these reports for a few minutes, I just want to give you a big picture view of what's in them. So our reports have pictures, they have maps, they have graphs with the data, and then they have some text for explanation. And the nice thing about the design and the setup that I'm going to show you is that you can generate all of these things together in a template and then iterate on that using different data sets. So it can give you a nice consistent result time and time again at the press of a button. And this is where it's time to introduce the tools that I used to make the reports. So I used a open source programming language called R, which is really common and starting to be used a lot in scientific analysis and in a lot of classes and statistical analysis. And then there's a framework or a template that you can create using R called an R Markdown document. And that's really that's the key for how I make these reports. So if we look at generally the different parts of this type of document, of an R Markdown document, there are 3 main parts. There's a header where you do some document setup. It's not actually displayed in your final document. There are chunks of code, which is where you would do your data analysis of reading in your data and performing some calculations and making a map. And then you have some text, which is exactly like it sounds. It's where you provide the narrative part of your report. So by combining these 3 things, you can create a template for your report. And the nice thing about it is that you set up the template once, and then you have many different output options. So you can create a PDF, you can create HTML, which is essentially ready for the web. You can create Word documents if you are wanting to output to Word and then do something in Word beyond that. So there's lots of nice options, and it all comes from a single source. And so it really saves you a lot of time once you've set up the template. So for the farmer reports I'm going to talk about today, you can really break down the pieces into 4 main sections that I had to develop in order to get these reports finished. The first part is data. The second part are scripts, and that's just a term, that's a small program that you write to deal with your data and create this analysis. You have the template, which is our Markdown document itself. And then you have a special type of script, so a special type of program that allows you to automate that process and go through and analyze your data and create these graphs based on the variables of interest to you. So in my case, I knew I had 27 different farmers and I needed to create a report for each of them and do it automatically. So I'll explain how each of these kind of fits together. Data is something that I know you've all seen before. So this is just a CSV or Excel spreadsheet, either one works. And you start with your data. So this is an example of some of my data here. And then to process the data, rather than doing it in Excel, you process it by writing a script. So this is a short example of one of my scripts, and you can download all of my scripts actually at that link if you'd like to look at them. And so this serves multiple purposes. It first reads in your data set, and then you perform some calculations, and then finally at the bottom there's some code that makes the graph itself. So all of this is contained within the script, and it allows you to set up that one script and function that will work on the data for all of your different graphs. The scripts are brought together then within this broader template. So earlier I talked about the R Markdown template and the different parts that it has. And this is where you really get to set up how your document looks and the order of the pages and the pictures, the maps, and the graphs that go into it. And this brings it all together for your final output. So when you have your data and you've decided how you're going to analyze your data and make your graphs and create some narrative around that with the text, that is where the template really helps you. And just as a quick look at what part of my template looks like so you can see this, there are sections of code like we talked about with earlier in the R Markdown document. So in this particular example, I have a section of code that calls the script that I wrote, that little program to create my graph. And then I call that script to make the graph here in the document, and you can see the example of the soil organic matter graph. And a good thing to note is that even though you can see a combination of code here and the graph, in the final report, you only see the output. So you get a nice polished version, but right now we're looking at the code. So this has not yet been turned into that final PDF report. Which really brings us to the question, how do we get from this document with these different chunks and the code and the data and the setup to a polished PDF? And that's really where the R Markdown as a tool, I think, really shines, is because you don't have to worry about setting up the size of your graph or your margins or all of that tweaking. You do it once in the template, and then every time you run your template, you get an output in your specified format. So for me, it's a PDF, and everything is always going to be the same size and setup here. So the way that I chose to get from my template to a PDF was actually to write another small script that I'm calling a render script here. So again, just a short program. It's a short set of instructions that tells your computer exactly how you want to take the data and your analysis and put it together in the template and then have a finished product. So this is really the key to the automation part of my talk that I was talking about. So this automation lets your computer do the really repetitive stuff that computers are good at doing, and it allows you to define the variables that are part of your report and let your computer create all of your different reports at the push of a button. So in our case, we just create a list of our 27 farmers, and as an output, we get a list of 27 different reports saved to the computer. And what this looks like in practice is actually really simple. It's about 20-ish lines of code that I'm showing here. And even if you don't read code or aren't familiar with code, I think you'll be able to understand exactly what happens here just with a short explanation. And I think it's helpful just to see the different steps. So we start just by creating a list. In my case, I have farmer names and region names that are part of my data set, and that's what I use to subset my data and do my analysis. So I create a list. And then I define the different variables that I'm using. So I'm using farmer names, and along with those farmer names, there's regional data that I want to pull in. And then I need to define exactly the output type. So in what is the file going to be called? Where is it going to be saved? And lastly, for the automation, I have a function here that looks at every line of the list that I made, and it makes a report for every line on that list. So for every farmer, a report is made and saved according to that name, and the whole process takes about 4 minutes. So it's pretty amazing once you get it set up how quickly you can create a whole set of these reports all at once. Now that we've talked about sort of the guts of this and how it fits together, I'll just do a brief tour through what the finished report looks like. And I hope that that'll jumpstart some of our discussion about different valuable ways that we can present this data and think about sharing what we know about soil health indicators with farmers. So we started just with a simple letter. You can see there's a map here, and there's a table that lists the different activities that we plan to do for research in the future, just to give folks an idea of what's coming up. We have a section where we talk about the research design and show a little picture of their field and where we took the samples. And then we also just generally talk about why we chose to sample where we did and the differences between those areas. And then we get into some of the data. So for each indicator, we show a graph, and then we have a short explanation for what that indicator is and how we understand it to relate to soil function. So soil organic matter, this is percent loss on ignition method, is pretty familiar to most people. And you'll see as I run through the different pages here with the different indicators, the decision that we made for how to display this data at this stage when we just have a subset of the total indicators is we wanted to show people the data for their farm. So that's in sort of a pinkish red color, and showing the mean in the middle with that black labeled dot. And then the colored dots actually show the full spread of the data. So we have error bars that represent the standard deviation there. So this is kind of a coarse view, but it just gives people an idea. What did we see on your farm? What did we see on regional farms? And then how does that compare to what we see in regional natural areas, just to put that data in perspective. And you'll also notice that this graph has two different panels, and at the top they're labeled A, upper, and B, lower. And this goes back to our research design, where in every field we were interested in finding an upper hill slope position, more of a shoulder summit position, and a lower position on like the back slope or the foot slope to compare within the field, how does that hill slope impact the results that we're seeing? So we show most of our graphs split into these two panels just to illustrate that sometimes there are pretty big differences just within that one field based on where you pull your sample. So moving on, soil pH, just a little reminder at the top of pH levels, and then down to the data at the bottom showing differences in pH, again, between those upper and lower samples. We have phosphorus and potassium data, and I'm not spending a lot of time on these. You know, you're welcome to read the report in full if you're interested in the text, but what I just want to point out here is our approach to sharing this data, especially for phosphorus and potassium. You'll see that I have colored shading on the graph that shows high, medium, and low. And we chose to do that because we have, you know, in Minnesota and most states, you have relative nutrient levels for your state and your soils that are generally agreed upon in these very low, medium, and high categories. So we wanted to put this on the graph because we thought people would appreciate being able to just glance and see, what does this number mean in the grand scheme of things if I'm trying to make relative comparisons? And that's information that we have for these standard nutrient tests, but I think the big point and the reason why we're all doing research on soil health indicators is that we don't necessarily have a great read or understanding yet on what high, medium, or low means for some of these soil health indicators, and how do we go about interpreting that and making that relevant to the specific context of the soil type and the type of sampling and all of these different factors and types of variabilities that we see. So you'll notice for these next couple graphs, I do not have the shading. So we have this 24-hour carbon mineralization or soil respiration measurement and potentially mineralizable nitrogen. So I think there's still a lot of interesting data to be gleaned from these graphs, and that's one of the reasons why we chose to separate out the your farm data from regional farms and natural areas, is just to show relative differences at this point, because we are still learning about how exactly we make the direct connection between the numerical soil health indicators here and then the types of outcomes and questions that people want to know about how quickly this is changing and the differences between management. And then the final page of the report, this is very simple, is just a list of contact information that we have and some acknowledgements at the bottom. So having said all this, I'll just share a couple of the questions that we tend to ask ourselves in our research group and that drive us as we're collecting this data and thinking about how we go out and share it and interpret it. We are curious, how do we think about interpreting these indicator results for people? And how do we get to a point where these indicators that we're measuring, which ones are going to be the most relevant and be most of interest to our stakeholders? And that's one of the reasons why we wanted to put the information out there early and get some feedback. And then a second question that we think about is, what does it, this high, medium, and low approach, is that appropriate for soil health indicators? We certainly have this information thanks to decades upon decades of research for standard nutrient tests, but I think there's a good argument to be made that some of these soil health indicator tests are pretty different in nature to the standard nutrient tests, and we don't have enough data at this point to really say what high, medium, and low are for most things, for most of these newer soil health indicators. So we're still learning that as we get an understanding of the variability and the differences based on the context. And then finally, what's the value of comparison between farmers that are using different practices, between management practices, and in reference to this natural area? So for us, we're trying to capture many different types of comparisons within the data, because I think that there's value to each of these and different sort of information that you can glean from those comparisons. So with that, I'll say thank you and look forward to answering questions. And on this slide, you'll see I have contact information for myself and Dr. Anna Cates. And then I have copied again that link. If you would like to go back through and take a look at the reports or check out some of the code, you're welcome to go there and download it.